Hello, this is Basil Al-Shakhdib, and I'm going to present here this poster as a part of a project called Fair Clinical and titled as Structuring a Clinical Case Report Form into OMOP Common Data Model. This project is a collaborative effort that includes different European universities and institutions, including the University of Luxembourg, University of Nottingham, University of Paris-Saclay, and the Swiss Institute of Bioinformatics. So, Fair Clinical aims to handle a very important aspect in the clinical publications, which is dealing with the supplementary data files. In fact, the supplementary data files in the clinical domain might contain a very important information, which is the clinical case report forms. Such reports are describing the diagnosis, treatment, symptoms, or even the test result of a particular patient. And given the challenge of searching and retrieving result out of those data files, supplementary data files, due to their heterogeneous nature, because they are coming from different data formats, including PDF, MS Word, PowerPoint, or even spreadsheets. So Fair Clinical aims to enhance the verification, findability, accessibility, interoperability, and reusability out of those supplementary data files. To do so, Fair Clinical is going to leverage the state-of-the-art pre-training language models in order to perform, firstly, the biomedical named entity recognition, normalization, and then identifying potential relations among the entities. Then, Fair Clinical is going to standardize the result of extracted such artifacts into a common data model, specifically the OMOP format. On the other hand, Fair Clinical also is going to focus on a subset of the CRFs that have been identified out of the supplementary data files in order to create a synthetic or analogon of the electronic health record. Let's take a look at the structure of the project. As shown in Figure 1, the project is composed of four main phases. The first phase is the data gathering and standardization. The second phase is the named entity recognition and normalization. The third phase is the relation extraction and OMOP population. And finally, the fourth phase is the indexing and updating of the enriched contents. The first phase, which is the data gathering and standardization, has been led by our UK partner from the University of Nottingham, in which they have performed a search query using the keyword case report through the PubMed Central, and they have retrieved more than 100,000 of full text publications, along with a couple of thousand of supplementary data files. Then they have standardized those supplementary data files into a machine-readable format of the BioC format, which enables the annotations and the enrichment of metadata. The second phase, which is the named entity recognition, which has been led by our French partner from the University of Paris-Saclay, is intended to identify the biomedical concept within the BioC of the supplementary data files that have been provided from the first phase. So, the aim here is to extract such biomedical entities and grounding them in a process called entity linking, in which the popular relevant database and ontologies regarding those entities are being brought to extract the original identifiers of those entities. So we are speaking here the MISH database for the disease and the diseases and the population entities, the UniProt for the proteins and gene entities, the KB database for the chemical compound, and so on. And currently, our French partner are benchmarking different BERT models in order to identify the most accurate model in terms of the extraction. And they have started with a PicoBERT model, which is intended to identify the population, the intervention, comparison, and outcome entities. And then they will provide the results of such extraction also in a BioC format, the same BioC format that have been initially created by the first phase is going to be used also here, in which the extraction of those entities along with their identifiers can be also enriched in the meta information within the BioC format. The third phase, which is the relation extraction, is led by our university, the University of Luxembourg. And this phase is expecting to receive the BioC, the enriched BioC format from our French partner in the second phase, where the biomedical entities and their identifiers tags are being depicted within the BioC format of the supplementary data files. And here this phase is intended to identify potential relations among these entities. We're here talking about the drug-drug interaction, protein-protein interaction, gene-induced disease, chemical-induced disease, and adverse drug reactions. And currently we are benchmarking different BERT models, including the BioBERT and the PubMed BERT models, in order to identify the best result and performance in terms of the relation extraction. And as soon as the results are being achieved, this phase is also intended to standardize the enriched contents of the biomedical entities and the relations within a common data model of the OMOP format, in order to create the synthetic or the analogon of the electronic health record. Finally, the fourth phase, which is indexing and updating the enriched contents, is led by our Swiss partner, the Swiss Institute of Bioinformatics, in which they are expecting to receive the enriched BioC format of the supplementary data files with the new annotations of the biomedical entities provided by the second phase and the new annotations of the relations provided by the third phase. Then they are going to integrate, update, and index the new content into their SIB literature services. They are currently developing a new framework that is able to handle different annotations, annotation structures, and digest them in a unique and uniform, compatible way of annotations. And that's it about FIRE Clinical. Thank you for listening.